Yes, Maniba and Anisa, you people also switch on your video. Everyone means everyone. All right, so in front of you, you see few questions from the biotechnology and its application chapter. So first, you people try to attempt these questions. Then we'll discuss these questions to see whether or not the topic or the concept is clear to you. And Aisha, you have asked me to revise molecular basis for you, right? Did you study
Yes,
that
ma'am.
chapter?
Yes, ma'am.
Is there any
Like how to
topic that
treat.
you have a doubt in? Hmm.
Yes, ma'am.
Which topic?
The rough R strain and the rat experiment.
The experiment of the mice one, the S strain and
Yeah,
the R strain. Okay.
R
Okay,
strain.
we'll discuss it. You people first complete this question, then I'll discuss that topic. One second, students. This thing has gotten stuck. Just one second. Done. Everyone's done till here? Shall I scroll down? Everyone's done? Okay.
Yes, ma'am.
This fourth question is also done. Done, students. Shall I scroll down?
No, ma'am.
Okay. Done.
Done, ma'am.
Okay, okay. People solve.
Done.
Everyone's done. Shall I scroll down? Can you show the 10th question? Done. Everyone's done till here. Shall I scroll down?
Yes.
Okay. Ma'am, can you scroll down? Everyone's done? So let's go down. Okay, last two questions. Done, students? Yes, ma'am. Okay, everyone's done till here. Shall I not scroll down? Shall I discuss the questions?
Yes, ma'am.
Okay. BT cotton is not. Which of the following is it not? A GM plant, insect resistant, a bacterial gene expressing system, resistant to all pests. Which one is it not? D. D. Is everyone's answer D? Why?
Yes,
Because
A.
it is particular. Which one?
A.
A. No, it's 100% genetically modified crop. If they are asking which of the following is it not?
Okay.
So yes, it is not resistant to all the pesticides. And resistant to pesticides, what does that mean? Resistant to pest would be something. Pesticides are the chemicals that kill the pest. So why would it be resistant to the pesticides? Isn't it? The people are getting this question? Everyone understood why the answer is D? Okay, C peptide of humulin or human insulin is a part of mature insulin responsible for formation of disulfide bridges, removed during maturation of proinsulin to insulin, responsible for its biological activity. Which one is the answer?
C.
How many of you have opted C? Yes, C is the answer for it, because if you people remember that when we talk about human insulin, it is made up of three chains, A, B, and C. The C chain gets removed when the insulin has to become mature in order to perform its function. So yes, 100% the answer is C. GEAC stands for
Genetic Engineering Approval Committee.
Genetic Engineering Approval Committee, right? Okay, alpha antitrypsin, what is it?
An enzyme.
It's an enzyme. Try to be specific, students. Remember, what is it used to treat? It is used to treat emphysema. Okay, these were in the notes, okay? So it is used to treat the emphysema. Okay. A probe, which is a molecule used to locate the homologous sequence in a mixture of DNA or RNA, it could be. What would be the nature of probe, students?
Single stranded.
Which one?
C.
C. Either RNA or DNA.
Yeah.
How many have opted for C? No one else. Then what are your answers? Other than C? C.
C.
Okay. What about others? Which one? Neither of any. None of the above. Yes, C is the right answer. It could either be a RNA or it could be a DNA. Choose the correct option regarding retroviruses. It is an RNA virus that synthesizes DNA. It is a DNA virus that synthesizes RNA. It is a single-stranded DNA virus. It is a double-stranded RNA. What is the answer for this?
A.
A. It is an RNA virus that synthesizes DNA during infection. And can you tell me the name of the enzyme it uses to do so?
Reverse transcriptase.
Yes. Reverse transcriptase is the enzyme, right? And what is the phenomena associated with it? Do you people remember? Like there is central dogma. So for these viruses, what is the thing called as? Aisha, you too might have studied this. It is reverse central dogma, which is also called as demonism, depending upon the person that actually gave the note about it. Okay. Okay. The site of production of ADA in the body is
Lymphocytes.
lymphocyte. Particularly which one?
T cells and
T
B.
cells. T lymphocytes. Not B, T lymphocytes. Very good. A protoxin is, what is the meaning of protoxin? Like whenever, remember I told you if any word has pro before it, like pro hormone, protoxin, what is the meaning of that? Pro means pro, very good. Like that. Someone said something?
Inactive.
Inactive. Anything pro is inactive. So pro hormone, pro insulin, pro toxin, it means it's not working right now. It has to get activated in order to produce the effect. So that is inactive toxin. What is pathophysiology? Pathophysiology. This though is not even mentioned. I haven't even taught you. What do you think the answer is? Pathophysiology. So even if you want to do so, it's okay. And tell me one thing, students, you people are doing these questions very honestly, right? You're not looking out for chat GPT and internet, na? Okay. Because even if you are, it's not my loss, but still. So pathophysiology, it is a study of altered physiology of the host. Meaning, when there is any kind of disease that is caused in any host, whether it is human, whether it is any animal, so we study the pathology of that person referring to the like host's physiology. Physiology means the body functions. So that is what we call as pathophysiology. The trigger for activation of toxin of Bacillus thuringiensis is, what is the trigger?
Alkaline pH of gut.
Alkaline pH of gut. Whose gut?
The insect.
The insect's gut. That is why whenever the insect eats the particular plant or feeds on the plant, they get the endotoxin that gets activated. The protoxins become actual toxin, thereby causing great deal of perforation in the gut of the insect. All right. Golden rice. What is golden rice? A variety of rice grown along the Yellow River in China. Is it Yellow River? So yellow rice or golden rice? No. It is made up of gold. That's why golden rice. Imagine how expensive that would be. A transgenic rice having gene for beta carotene. Yes. And what is this particular rice enriched in? Like, why are we calling it golden rice and why is it so valuable? Anyone?
Vitamin A, it helps.
It has
It
a
prevents.
higher quantity of vitamin A as compared to the normal rice. Apart from this, the golden rice is yellowish, sort of yellowish in color, like how the normal rice is whitish, but this one is sort of yellowish. Okay, RNA interference genes are silenced using which of the following? What happens during RNA interference? See, normally in the cell or anywhere, there exists a single-stranded RNA. This is the normal case scenario. Why? Because this single-strand RNA will undergo the process of translation. Okay, it will undergo the process of translation and form what? After translation, what is formed? What is formed after translation? This is the simple question. After translation, what is synthesized? Proteins. Protein is made. Okay, so when this RNA has to undergo the process of translation, it has to be single-stranded. But if you make this RNA double-stranded, then the process of translation will not take place. Thereby, the protein will not be formed. So genes are going to be silenced. So it is mostly the double-stranded RNA. Okay, the first clinical gene therapy, it was done for the treatment of. Yesterday only people have studied this.
B.
Yes. Particularly it was given to a child, and how old was she? This is important. 7. No, she was 4 years old. Okay, she was 4 years old. She had ADA deficiency. So as to treat her, this clinical therapy, gene therapy was given. Okay, ADA, what is the full form of ADA?
B.
Arginine diamide. Huh? Which one?
B.
Adenosine diamide, right? ADA. Very good. Okay, silencing of a gene could be achieved through the use of one more. This is a single strand RNA. The RNA that will be synthesized against this will be called as the antisense RNA. Okay, so the silencing of the gene is done with the RNA interference mechanism also and antisense RNA. So this would be the answer. So these questions, how are they? Easy or difficult or impossible or very good? Or none of the above? You people were able to do so, right?
Yeah, ma'am.
Okay. All right, so let's come on to the revision of that particular topic. Aisha asked me to do so. That was transformation, right? The transforming principle. How many of you remember that topic? Do you people, like, who is confident enough to tell me what that thing is about? transforming principle. Safrina, you can?
Yes.
Okay. Explain it quickly, then I'll move on and tell you about it. Why was it done, first of all? It was done because there was a need to find what was the genetic material, right? No one knew back then that what exactly was the genetic material. So then the search for the genetic material began, and thereby to find out what in fact was the genetic material. Now tell me what happened during the transforming principle. Acha, first tell me who gave it. Anyone?
Frederick
Griff.
Frederick and Griffith. Everyone agrees? Everyone is agreeing to her. Okay. And now tell me what happened in it.
So he grew Streptococcus pneumoniae in a culture plate first,
Hmm.
uh, which produced S strain and R strain,
Hmm.
where S strain is virulent.
Hmm. And our strain is non-virulent. Yes, correct.
Yeah.
Next.
So he injected that into mice.
Hmm.
When he injected S strain into mice, the mice
Hmm.
died. And he went,
Hmm.
when he injected R strain, it did not die. It lived. So that was one point. And then he discovered that bacteria can be killed by heating. So he heated S strain first and then injected into the mice, and the mice lived.
Hmm.
Then he heated S strain heated with R strain,
Hmm.
and he injected that, and the mice died.
Hmm.
So he concluded that there was some transforming principle in the S strain that uh, transformed the R strain to kill the mice.
Ha. He made like something got out of the S strain and got into the R strain, making it virulent when it was non-virulent. Very good. Excellent. Very nicely you've explained all the points you've mentioned. Very good. And where are the rest of you? One by one, everyone switched off their videos? Switch it back on. Quick. One by one student went, another student went, another. What happened? You people doing train, playing train. Switch on your videos, everyone. Quickly. So talking about the transforming principle, it was given by Frederick Griffith. So in the year 1928, this person was working with Streptococcus pneumoniae bacteria. And he noticed how the different strains or the different colonies of this particular bacteria showed different kind of effects. So he was growing this Streptococcus pneumoniae bacteria, and this bacteria actually has two types of strains that it produces, S strain and R strain. S strain stands for smooth and R strain stands for rough. The S strain is smooth because it has a proper mucus coating or mucopolysaccharide present on it, which provides a virulence factor to that particular strain. Whereas the R strain does not have that mucopolysaccharide, as a result of which it is not virulent. So what he did, he took the S strain bacteria, injected into the mice, and he saw that the mice died, showing the fact that mice has gotten pneumonia, and because of that it died. Second, he took the R strain bacteria, injected into the mice, saw that the mice did not get any pneumonia and the mice lived. Then he heat killed the S strain and then inserted into the mice. And then he saw that, okay, because the S strain was killed, so the mice lived and it did not die. But much to his surprise, when he took the heat killed S strain, mixed it with the R strain, and then injected into the mice, now the mice died. So S strain, which was virulent, it was heat killed. And R strain was non-virulent, but still the mice died. So he made a speculation that somehow the R strain has been transformed into becoming S strain, and caused this thing, pneumonia in the mice, as a result of which the mice died. Later on, when he extracted the things out of the mice, he found S strain from the mice. So this concluded that there is some sort of transforming principle which gets, you can say, which transforms the non-virulent type of strain into the virulent type. Is it clear to everyone? Is it clear, Aisha? Okay. Yes, any other topic that you people had a doubt in from this chapter? Is there anything else, Aisha?
The lack of brain model.
Lacoperon. Okay. Now see, lacoperon refers to the lactose and its metabolism system, right? And when we talk about the operon of that organism, mostly that lacoperon is found in the prokaryotic organisms. Now, it has the inducer, it has the promoter area, it has the operator area, and there are structural genes present. Different structural genes give rise to the different kinds of proteins, ultimately forming the enzyme. So when we talk about the whole entire setup of the bacteria, so there is the lac Y, lac A. Now lac Z codes for the enzyme beta galactosidase. Lac Y codes for the enzyme transacetylase, and lac A codes for the enzyme permease. Now what happens is that, why are these enzymes present? These enzymes are present in order to metabolize the lactose, right? Because this is the lacoperon that we are talking about. So whenever the lactose, which is called as the inducer, when that is absent in the medium for that bacteria, so this, you can say, inhibitor or this particular set of operon is going to synthesize a repressor mRNA, which will get translated to form the repressor protein. Now, since there is no lactose present in the medium, so this repressor protein is going to go and bind with the operator, as a result of which RNA polymerase could not bind to the operator, and none of the structural genes are getting to get transcribed. No transcription happens, thereby no translation happens, and no enzymes are synthesized. So when no lactose is there, no enzymes are synthesized because the repressor is blocking the operator region. This is the case when the inducer is absent. Second, the presence of lactose or any form of lactose like allolactose, like that. So when inducer is present, now when the repressor protein will be synthesized, this this inducer, that is the lactose, when that will come and bind with the repressor, it will change the conformation of the repressor. Changing the conformation means if it is oval, it will make it circle. If it is circle, it will make it square, meaning it will change its shape. As a result of which, the inactive repressor will not be able to bind with the operator, and thereby the three enzymes, that is beta galactosidase, permease, and transacetylase, will be synthesized. Understood? So always, whenever you have to explain about the lacoperon, you have to give two case scenarios: when the lactose is present, when the lactose is not present. Then only your answer will be complete. Is it clear to everyone? Okay. Any other thing? Very good. Now, we have discussed this. We have discussed the questions also. Okay, so let me start a new chapter now. So the chapter we will be starting is evolution. So do you people know anything about evolution? How will you know? I haven't even taught you yet. Can you see the screen that I'm sharing? Everyone can see the screen?
Yes, ma'am.
Okay. Now see students, when we talk about evolution, first I would like to tell you what all things are present in this particular chapter. So when we talk about this chapter evolution, we'll see how the universe was formed, how the earth was formed, and then how life came into existence on earth. And then whatever life came into existence on earth, we will see how that life evolved, like how there was initially the unicellular organisms, which like from them the multicellular organisms came, and then higher organisms. We'll see how different kinds of theories of evolution has been given. Like, there are so many people, there are so many, you can say, researchers out there. There were also, and they are still present also. So everyone has, explained evolution according to what they think it is. And those became the theories of evolution. So we will study the various theories of evolution as to how evolution happened, what contributed to the evolution taking place, and the most interesting one, we will see how the man evolved, how there were cavemen, how the evolution of man took place. So it's this is the whole entire chapter. Now, whenever we talk about evolution, so talking about what it actually is, so it is a process that results in heritable changes in the population. The heritable changes in the population spread over many generations, leading to diversity on Earth. It tells us how the life form that existed earlier do not exist anymore and they have evolved. Being evolved means changing into something better or something new. So the study of the history of life form, we call it as evolutionary biology, right? That is like everything has a set of study that it requires. So the study of evolution and the history of how the life forms were, how they are now, how they became what they are today. It's because we study the whole entire this thing, evolution, that comes under evolutionary biology. Now talking about the origin of universe. Now universe is a collection of clusters of galaxies, and the galaxies contain the stars, it contains the clouds of gases and dust. And almost like approximately, they don't know it like as a fixed value, but approximately the universe is almost 20 billion years old. That's how old the universe is. 20 billion years old. Now the question comes along, how did the universe came into existence? Right? Since it is like we are studying something that universe was formed and everything. So how did it came to existence? So the origin of the universe is explained using this Big Bang theory. How many of you know this thing? Big Bang theory? Not the serial. There is a serial also that is called as Big Bang theory, but not the serial. I'm talking about this Big Bang theory that actually took place. Speak louder, I can't hear you. Say Aisha.
I know.
You know it. Everyone knows Big Bang theory? Or how many of you have heard it for the first time today? It's fine. When I studied this, I heard it for the first time. See, when we talk about the Big Bang theory, basically it tells us how the universe is very, very old. And initially, how the universe was formed, it's because of a single huge explosion that took place almost 20 billion so years ago. And then what happened is that the whole entire universe was formed as a result of the Big Bang theory. It was a single explosion that was huge. And due to this particular explosion, the gases were formed. Okay. And the temperature, like slowly hydrogen, helium gases like these were formed. Right. And because of that, the whole entire universe expanded and the temperature came cooling down. So early in universe, everything was condensed in a single small point of infinite denseness and heat. That's all there was. Then a huge explosion, meaning the Big Bang. So a huge explosion took place, that is Big Bang, and then the universe expanded, the temperature came down, hydrogen and helium were formed later on. The gases condensed under gravitation and formed the galaxies that we have today. The one that we live in is called as, which galaxy do we live in? Milky Way. How many of you have heard about Milky Way? The whole entire planet things that you see, the picture, that is one of our planet is also there, which is the Earth. So like all other galaxies, our galaxy, which was the Milky Way, was also formed. This galaxy contained our solar system, in which the Earth was formed about 4.5 billion years ago. Okay. Now, talking about the origin of the Earth's atmosphere, how the Earth's atmosphere was formed. So, in the Milky Way, there's the solar system, then comes our planet, that is the Earth. Now, there was not, there was basically no atmosphere present on the early Earth. None. Later on, what happens is that the ammonia, the methane, the carbon dioxide, and the water, it was released from the molten mass, meaning from the lava, and it started to cover the surface of the Earth. Later on, the water, under the effect of UV rays from the sun, it got split into hydrogen and oxygen. Now, this hydrogen is a lighter gas, so it escaped out. Talking about the oxygen, it combined with the ammonia, it combined with the methane, and then what happens is that when it combined with the ammonia, it formed the nitrogen. When it combined with the methane, it formed the carbon dioxide. So these gases came into existence like this. Later on, the the oxygen, under the effect of the UV rays from the sun, it got split into two molecules or two atoms of the oxygen. And when they combine, one of it combined with the oxygen, then we form the ozone. How many of you know the significance of ozone gas? See, this, like, in you people remember those things you might have studied: stratosphere, troposphere, atmosphere, all those spheres of life, the biosphere. Huh. So when we talk about the ozone, so it is like a layer of gas that is present in the lower stratosphere, which forms sort of like an envelope, sort of like a blanket around the Earth. What it does is that it does not let the harmful UV rays from the sun reach the Earth and thereby protect us from a lot of harm. So this ozone layer was formed later on. As it cooled, the water vapor condensed and fell in the form of rain. So wherever there were depressions on the planet, right? Depression means like, what do you call it? What do you call depression, students? Like holes and areas that were sort of, I don't know how to explain that. Like those areas that had some sort of hollowness in them. Those were the depressions, right? So then when the rain fell on the earth, those depressions got filled with the water. And then the ocean and the different water bodies were formed. Early atmosphere was reducing in nature. Okay, why? Because there was no oxygen left, which was suitable for life. But life forms existed 4 billion years ago. This is the main thing that you people must know. This much is clear, students. Do you people get the gist of it, what I'm trying to say?
Yes, ma'am.
Any questions? Any doubts?
No, ma'am.
Okay. So basically, life appeared. One more thing. Life appeared after these things were done. Then the first form of life appeared, and it was basically 500 million years after the formation of Earth. Or, all in all, you can say 4.5 billion years ago, life appeared after the formation of Earth. Is it clear? Okay. Now you people note down. Take a fresh page of your notebook, note these things down, and then we'll move forward.
Done. Done. Done.